I'm Dr. Shakthi and welcome to another episode. We'll be discussing about caveolar vesicular transport today, and it's gonna be a little more in depth. In case at any point you feel that this is too much for you, I put up another video. You can check out the link over there. It's a little more simplified version. So in case you have trouble with this, you can go check that out instead. Okay. Let's come to the membrane structure. So there's a particular microdomain in the membrane which is rich in cholesterol, sphingomyelin, glycosphingolipid, and this is what is called a lipid raft. So in these lipid rafts, there is presence of integral proteins like the caveolin. Caveolin 1 is there, 2 is there, 3 is there. Caveolin 1 is by far the most important. Caveolin 2 is also known to play a role. Cavin 3 has been recently discovered. So basically caveolin 1, 2, 3 seem to play a role, and caveolin 1 seems to be the most important. Now caveolin is this integral protein, and this together along with the lipid raft and the caveolin forms what is called the caveolae. So caveolae, an individual caveolae has around 144 molecules of caveolin. This caveolin structure, you can see that one side of it is attached to the lipid part of this. Okay, so that is palmitoylated. So that is your C terminal. Okay, the carboxy terminal has been palmitoylated, and at one point in the N terminal, this is the N terminal, and at one point in the N terminal, there is a scaffolding domain. And that scaffolding domain helps it attach itself to your cholesterol and sphingomyelin rich areas. Apart from all this, there's one particular segment here near the N terminal where tyrosine residues are there which can be phosphorylated, and this is very important later for its activation and all that. That is your caveolin. Now in this whole lipid raft region, the presence of phosphoinositol, and that phosphoinositol gets glycosylated, and anchored to it is a protein. So that is your GPI anchored protein. Okay, so that is glycosylated phosphoinositol anchored protein. Okay, so this is your whole caveolar structure, and this is important for signaling. So activation of this will help in signaling. So this again is membrane associated protein. You can check out the link if you want to understand a little more about these. I already discussed it in a previous lecture. Now beneath all this, cytoskeletal elements are present, for example actin. Okay, so this caveolin is attached to cytoskeletal structures like the actin through filament. Okay, and apart from all this, there is a peripheral protein which is involved called the cavin. Okay, this cavin also, just like this, has 1, 2, 3, and each has its own role to play. Okay, that unfortunately is beyond the scope of this whole lecture. There are a lot of details about that. Okay, so this has a trimeric formation like this, and that can oligomerize. More of these can combine together and can oligomerize. So this kind of, what it does is, if you already know about clathrin and clathrin cage, something similar to that, this cavin helps form a structure around what is being internalized. Okay, and each 1, 2, 3 has its own separate, very distinct use in the whole sequence of activity. Now, in regard to what actually activates this are ligands such as this. Okay, so folic acid and albumin is a lot of studies have been done, and we come to the understanding that the endocytic pathway in this is through caveolin and all that. Apart from that, autocrine mobility factor, alkaline phosphatase, lactose, ceramide also have caveolar pathways associated. Pathogens such as all these examples are also known to play a role. So a lot of studies have been done, especially on SV40 virus. Okay, so a lot of things we know about caveolar activity is through studies done through this virus. Now coming to the whole mechanism of how it acts. So we know that the caveolin is there. Caveolin 1 is the most important. ligand binds, and through this whole signaling pathway that is there, it's going to go activate a few things. Okay, first thing you have to understand is something called the SRC kinase, that usually its activity results in tyrosine phosphorylation, and we already know that there's one point over here near the N-terminal which is for tyrosine phosphorylation. Okay, there's a tyrosine residue over there, and this SRC kinase does that. But normally there are PP1, PP2A, these are phosphatase. Okay, so these phosphatase are, serine, threonine phosphatase, and what they do is normally inhibit your SRC kinase. And by the activation of all this through ocadaic acid, they are going to inhibit the inhibition over SRC kinase. So what happens eventually is SRC kinase is going to tyrosine phosphorylate it, activating the whole mechanism. Okay, and your dynamin structure also comes over here. Okay, so over here it is what is called the dynamin 2, and this dynamin 2 is recruited with the help of nostrin. So nostrin is epithelial nitric oxide, so you must be knowing that. Okay, so nitric oxide synthetase, epithelial nitric oxide synthase. So epithelial nitric oxide synthase trafficking inducer, that is nostrin, and with the help of that it recruits dynamin 2, and dynamin 2 acts like a spring. GTP hydrolysis acts like a spring, and it's going to help it internalize. So a recap of this can be seen over here: cholesterol enriched region, the lipid raft, and how a ligand comes over here. This is a virus, and through caveolin you can see the cavin 1 here, and that results in internalization through pinching off by dynamin, and finally this whole thing is going out. What you have to understand is this whole complex that is being formed has NSF, SNAP, VAMP, and these are things that help in docking and fusing. Okay, so as when we're talking about endocytosis, with regard to that, it is going to help it fuse and dock with cytoplasmic organelles. Okay, you might be otherwise knowing about all these things with regard to how exocytosis happens. And this again is something we have already done, this whole pathway over here. So this finally phosphorylates it, the phosphate group is added, dynamin internalization. After that, the next step requires dephosphorylation. Okay, so the phosphate groups are removed, dephosphorylation, that is recycled, and then comes the next step. So in this whole process, after this, there is reorganization of the cytoskeletal structure. So that is what is going to help in internalization. So I hope you remember the filament and the actin which is involved initially, and reorganization of that cytoskeletal structure will help it internalize it. Okay, and after that, it's going to propel with the help of motor molecules along your microtubules and gonna keep going inside. Okay, so this caveolin forming the caveolae, and that caveolar complex, one of that binds to another one, and multiple ones keep joining to form what is called the caviosome. So you have to understand that caviosome is not like endosome, it's kind of a little different. Okay, so caviosome is a multi-caveolar complex, often referred to as grape-shaped, sometimes even star-shaped. Okay, they have a neutral pH. Unlike endosomes, over there is acidification. Okay, they fuse with lysosomes as well. Okay, and these caviosomes are very distinct from your classical endocytic compartments like the endosome. But please do note that studies have shown that there's some sort of communication between the caviosome and parts of classical endocytic compartments, especially early endosome. There are a few studies showing some late endosomal communication as well, but more important was the early endosome communication. So over here you can check out the uh, grape-shaped caviosome here as well and here as well. Okay, now you can compare all these things. So these are phagocytic pathways and these are non-phagocytic pathways, and in the non-phagocytic pathway you can see clathrin mediated endocytosis, and over here is your caveolar complex. Okay, finally how caviosomes are formed. And you can see that this is not related. Okay, maybe some sort of communication is there initially, but ultimately this does not, combine with lysosome, do not form the multivesicular bodies and all that. Okay, after this, what happens next? Endoplasmic reticulum, Golgi, whatever happens, that depends on a few things. Okay, so as you can see over here, the caveolin 1, dynamin 2, cytoskeletal structure, cavin, all that is going to help in forming the caviosome that we saw. Okay, and from there what happens? That will depend on things like small GTPase, like the RAB, kinase, phosphatase. Based on what is there, the whole thing is decided based on that. Okay, for example, RAB5 means it's going to go into the early endosome. Okay, uh, presence of RAB11, PP2A, these phosphatase will make it go into the endoplasmic reticulum. Okay, and the RAB7, PP2A will make it end up in the multivesicular body. Okay, so based on all this, the final direction of it is determined by things like your small GTPs like the RAB, the kinase, phosphatase. Okay, so I hope this was clear. A major chunk of it was referenced from this one article. It's a good article. There'll be some extra points if you want in it, and of course from Boron's medical physiology. Okay, so thank you.